is Zachary Hancock, a postdoc researcher in our department working with Dr. Gideon Bradford. And Zach received his PhD in ecology and evolutionary biology at Texas A&M in 2020. He was then at Michigan State University working with Gideon until 2022 when the lab moved here with us. Zach's research centers on molecular population genetics and phylogenetics, and he's especially interested in how spatial and temporal structure impacts coalescence and how this together affects phylogenetic interference. He also has interest in mismatch between organismal complexity and genome size and in invertebrate systematics and phylogeography. So today we'll hear from him on amphipods in space, how spatial structure shapes genetic diversity.
All right, thank you everyone for being here. Thank you so much for the introduction. As she said, I'm a postdoc. Thanks for the introduction. Thanks everybody for being here and for your patience. As I said, I'm a postdoc in the lab of Gideon Bradford, where you might know we study spatial population genetics. So I'm going to talk a little bit about that at the end of the talk, but I'm also going to be talking a little bit about some of my PhD work. But how I really want to begin this talk is by convincing you just how cool amphipod crustaceans are. So the very beginning of this is going to be kind of a propaganda campaign to convince you of how cool they are. So, and of course, I have to click this. Okay, so there's around 10,000 described species of amphipod, and there's estimated about 25,000 in existence. And amphipods are paracharid, so these are brooding crustaceans, which means that they don't have a pelagic larval form. Instead, they actually just brood their offspring through several molt cycles, so they're generally kind of dispersal limited. They're sister taxa to isopods, which are roly-polies, as well as like the deep-sea Bathynomus. They're abundant in all freshwater and marine ecosystems. Now we're just seeing the little thing on the bottom here. Oh well. Okay, what was I saying? Amphipods. Lots of amphipods. They're really cool. We're seeing comments, so can you not comment? They're popping up on the big screen. So anyway, yeah. So, all right. One thing I want to talk about on this slide still is the mating strategies of amphipods are really cool. So the way that males they'll go out, search for females, and then they'll grab onto them and hold onto them until they molt. And this is called mate guarding. And so they protect them, swim around with them, feed them, take care of them, because the female will not pass her oocytes until right after she molts. And so this has led to this really interesting mate guarding strategy. So to kind of get a grip on just how diverse they are, we can start off with sort of a basic gamma-rid body plan, which is this one here. And then this is an example of that in Gammarus mucronatus. Is a very common brackish water amphipod, and then you can see all the different ways in which they have dramatically modified this basic body plan. Whether you're the whale louse Siamus or the skeleton shrimp and the coprelids, where you've basically lost all of your legs, and then you've got this twig-like body structure, or you're the cystoma up here, where what looks like it should be a head is actually an eyeball. It's just a gigantic sessile eye. So a great deal of morphological diversity in amphipods. These are some of my favorite ones. So this is an Antarctic epimeria. Has really pretty spine patterns as well as these red stripes. Here's a couple of other ones that I think are really dazzling. So this looks like it should be an extension of the back, but this is actually the first coxa. So it's the first article of the leg, and it's extended down over the rest of the legs in a kind of like shoulder pad structure. So really neat. And then here's a couple of other ones. You might be familiar with the Lake Baikal radiation. So this is a Acanthogammarus from Lake Baikal. You can see they've modified their back legs into these kind of like spider-like structures. So really cool. Down here are deep-sea Lycianacid amphipods that you can see they're really, really big. Most amphipods aren't this large, but in the deep sea, anything goes. And then lastly, I wanted to show you Phronema. This is one of the few parasitic amphipods. So this little guy parasitizes gelatinous animals, so siphonophores as well as planktonic tunicates, which are called salps. And what they basically do is they invade their little gelatinous body, consume all of their internal organs, and then just swim around with their gelatinous body as like a little house. And so then they just kind of poke their head out and feed as they're swimming around in their gelatinous body. Okay, so now that I've introduced you to the diversity of amphipods and we've played on Zoom for a while, I'm going to kind of lay out the three trajectories that we're going to go in this talk. First, I'm going to give you a sort of lightning round into the origin and evolution of a specific family of amphipods. I'm going to throw a lot of information at you to try to cover the breadth of things that we've studied and maybe give you an idea of things that we still have left to do. And then I'm going to talk a bit about spatial structure. And a lot of this spatial structure work comes from the things that we've learned in this first chapter. So, to introduce you to hosteriid amphipods, so these are a family of sandy beach amphipods. You collect them on pristine, open, sandy beaches. So this was one of my field sites in Grayton Beach, Florida. This was a really nice organism to work on because it basically just meant throughout grad school I got to hop from beach to beach. And you can collect these very easily. There's hundreds of them in every centimeter full of sand. If you've ever gone down to the beach and just let your toes sink into the sand, these things are crawling all over them. They're everywhere. They are fossorial, as I said. They're highly morphologically specialized to burrowing in the sand, but they are generally restricted to the northern hemisphere. So you won't find them in the tropics and you won't find them in the southern hemisphere. And they were first extensively studied by Ed Bousfield and J. Lawrence Bernard in the 60s and 70s. So to see how they've kind of modified that basic gammarid form, here's our gammarus with the very thin legs and the long antennae. And what the hosteriids have done is they broadly expanded those legs, and those long antennae are now much shorter and more robust. Also notice that this is its eye, so it's completely white. There's no pigment. They're completely blind. And then if we zoom in on one of the legs here, I've just kind of outlined this kind of cup-like structure. It's almost like a little shovel. You can see it's got a lot of spines and seedy around it, and they use that for digging. So their legs are highly modified into almost shovel-like structures. So in terms of the biogeography of them, they were first studied by Ed Bousfield, as I said, in 1970. And the way that amphipodologists worked on biogeography in the 1970s was the same way they did it in the 1870s, which was basically just a verbal argument based on shared patterns of morphology. And so effectively, this is a study from this paper, and he argued that they represented a very recent rapid radiation, that the center of hosteriid diversity was in the North Atlantic, and that they very recently colonized the Gulf of Mexico. And again, this argument was just based on morphological patterns. However, at the time, there were very few species recognized in the Gulf of Mexico. It hadn't been extensively studied, and so it's very hard to know if there was actually any fewer species there than there was in the North Atlantic where he was working. And when I started grad school, there was only one species that had any molecular data at all, and that was Hostorius arenarius, which is solely a European species. So setting out in grad school, what I wanted to do first is just go to all of the different sites in which these things had been reported. So that took me all across the Gulf of Mexico, as well as all of the North Atlantic to Prince Edward Island, just hopping from beach to beach collecting amphipods. And initially, my very first project was to describe 2 new species in the Gulf of Mexico, which was very rewarding. But I will say that I've done a lot of things since then: genetics, genomics, statistical modeling, etc. Nothing is as difficult as alpha taxonomy of hosteriid amphipods. Dissecting each one of these little legs and the mouth parts and the spines and the seedy was quite challenging. So I highly recommend everybody do some alpha taxonomy at some point in their life. Okay, so what I want to do now is show you some sequence data. So also on top of doing the morphological work, we also just basically sequenced everything that we came across. And we sequenced them for the barcoding genes, right? Because there was basically no genetic data at all. So we sequenced around 100 individuals from across the Gulf and the North Atlantic. And this included 20 new species that had never been sequenced before. And we uncovered extensive cryptic diversity. So what I want to do is show you this phylogeny. This is just from the species in the Gulf of Mexico. This little purple bar here, this is for morphology. So there's only 4 morphological species pictured in this entire phylogeny. And so as you can very clearly see here, there's a lot of cryptic diversity in this group. And this is very clear when we look at the Lepidactylus clade down here. So Lepidactylus triarticulatus is the only member of its genus represented in the Gulf of Mexico. This was all one morphological species. And you can see there it's deeply diverged as the middle Miocene. So there's a lot of cryptic diversity here. Furthermore, this was one species here where the red and the blue represent the North Texas and the South Texas coast. And the break between those 2 clades is only about 100 kilometers. So there's something that's completely dividing this single population into 2 reciprocally monophyletic groups. Finally, the major impact on the Gulf of Mexico, as you might would expect, is the Mississippi River. So the red stars here represent breaks on either side of the Mississippi, and they all show very deep patterns of divergence, indicating its importance in their biogeography. Okay, so the next I wanted to get into a little bit of Hosteria genomics, and my initial inclination was to do like some reduced representation sequencing, some like RAD-seq, to do some PopGen stuff. But when I went to the sequencing center, they said I needed some kind of estimate of their genome size. And at the time, I went to the animal genome size database and found that it listed amphipods from 500 megabases to 50 gigabases. And somewhere in that gigantic range were Hosteria amphipods, and I didn't know where. So I teamed up with J. Spencer Johnston to do some flow cytometry to estimate genome sizes, and they had a very incredible range. So from 2 gigabases at the smallest end, this is about the size of a chicken's genome, to 14 gigabases at the larger end. This is about the size of an onion's genome. And for reference, humans have a 3.5 gigabase genome. So the largest of these amphipods had a genome several times larger than our own genome. So we got really excited about this, and we wondered what the heck is like in the genome of these organisms that are so gigantic. So we did some low-coverage Illumina sequencing to kind of characterize the repetitive content within these genomes. And we found that upwards of 70% of some species' genomes were repeat content. And this was mostly dominated by DNA transposons. And these are the line elements, so long interspersed nuclear elements, which could be as much as 30% of their genome. And we did find other ones as well, LTR and non-LTR retrotransposons, and as well as satellite DNA, but these ones were the ones that really stood out. And they were strongly correlated with genome size. So the more repeat content you had, the bigger your genome was. And we also found an interesting pattern in that body size and temperature also strongly predicted genome size, which is a pattern that had been seen in other ectotherms as well. Okay, so lastly, we were kind of interested in like where the Hosteriids fit in the broader amphipod phylogeny. And so we used a fossil calibrated tree. There's not a ton of amphipod fossils. They don't fossilize very well, but there are a couple, so we were able to time calibrate this tree. So this represents 100 species across 30 different families. And one of the first things that kind of jump out at you is that there's as much diversity in the Hosteriids as there is in the entire Gammaroids. And this includes multiple families of all the Gammarids as well as their close relatives. So you can see there's an incredible amount of diversity in terms of how deeply diverged the Hosteriid groups are to one another. We also estimated they originated during the Eocene, so they've been around for greater than 40 million years. And we didn't find any evidence that all the burrowing amphipod families were a unique clade. So this might be a little esoteric, but it was a decade-long dispute that all burrowing amphipods should be monophyletic within a single group, and we didn't find that at all. They were spread all across the tree, and there was just no evidence for there being any discrete grouping there. So the last thing I want to show you from the empirical data are these two plots, and these are something that you might could have intuited just from sort of the life history of amphipods. So they're brooding crustaceans, and Hosteriids specifically are burrowers, and they're blind. So they're not very good dispersers, right? And so what you find in them are very strong patterns of isolation by distance. So what this table here is representing is pairwise FST across each of our sample sites, with the darker blue representing the greater levels of differentiation. So you can see very strong patterns of isolation by distance. Similarly over here, this is just for one species. It's a pairwise comparison across individuals. And again, you can see a very strong positive correlation with distance and divergence. So when I first presented this data, I included this figure to the Department of Biology at Texas A&M, and this was supposed to represent sort of like a hypothetical biogeography for the emergence of the different species on either side of the Gulf. And this led to Heath Blackman, who is a professor in biology, to ask a question about how ancestral patterns of isolation by distance might be impacting my divergence time estimates. So to understand exactly like what he meant by this question, let's zoom into this. So I proposed that ancestrally there was a very large range where the amphipods were spread all over the Gulf. And so since we know that isolation by distance characterizes the present population, it's probably true that they had isolation by distance here as well, such that those at the ends were probably much more diverged from one another than those nearer to where the Mississippi River is, right? So then the Mississippi River comes down, divides these populations, and then they continue diverging until the present day. And now what we want to do is go and ask, when did this happen? When did the Mississippi come and separate this population? Well, recognizing that there's some level of ancestral isolation by distance, you could imagine if you sampled right on either side of the Mississippi, you might get a completely different date than if you sampled at the ends of the range. Now, this seems pretty obvious in the Gulf of Mexico and the Mississippi River as a very clear, obvious vicariant zone, but for a lot of organisms, we don't know where that vicariant zone is. And it may not be as obvious as the Mississippi, and it may not even be present anymore. And so what I did is I kind of dug into the literature to see if I could figure out if there had been any work on this before. And as a crustacean systematist, I didn't have a lot of pop-gen knowledge. So I came across this paper by John Wakely in 2000, and I want to kind of walk through the logic that he lays out here. So starting off, this is pairwise divergence between species I and J. This is what we actually measure between those two species. And it's related to, obviously, mu, which is the mutation rate, multiplied by these two terms. Now, T sub d is the actual time of divergence. So this is when the Mississippi River came down, divided the population, and how much time it's been since then. 2 N of E, however, is the average pairwise time to coalescence within a population. So this is the number that we want. This is the value that we want. This is kind of a nuisance number. And to get an idea about what this means, so here are our two populations. We trace those lineages back to the ancestral population, so that's the time of divergence. But then it takes some time, once those two lineages are in the common population, to then find ancestors, right? And that takes, on average, 2 N of E generations. Now, what spatial structure does is it inflates this number. And it does this because once you're in that ancestral population, if there's low levels of migration, you have to wait for migrants to get within the same subpopulation so that you can have this sort of coalescence in the first place. So it makes this value get bigger and bigger. And so we asked, how might this manifest in subpopulations that are near that vicariant zone versus those that are very far from it? So we set up a simple simulation model, and I'm going to walk you through it. So starting here at the 1 on the tree, this is where we begin with just like a very large ancestral population. We allow it to evolve to reach some like equilibrium level of diversity. And then we split that population at the dotted line. We're along the 3, this is just our outgroup that we use to root the tree. And then along the 2 here, we take that population and we subdivide it into what we call a linear stepping stone model, where each one of these circles represent randomly mating deans with some migration rate between them, right? And so we allow this to go for 50,000 generations to establish isolation by distance patterns, and then we split it again. And we split it here for various values of like, just like the time of divergence. Now we split it in 2 different ways. One in which we allow just the ends to survive, and then we estimate divergence between them, versus when we allow just the centers to survive and estimate divergence between them. So the simulation puts out FASTA files, which are just like sequence files, and then we fed those into a popular phylogenetic program to let it estimate divergence times and then see which ones were deeper in time. Okay, so I've left this up here for reference, and I'm going to walk you through what these figures are going to look like. So for each one of the columns, you're going to see ratios of the time of divergence over the population size. For each one of the rows are going to be different migration rates, starting high and then going low. The y-axis is how close the estimated was to the true. So if they're along the dotted line, then they're right where the actual true value is. If they're above it, then they're overestimating the true value. So this is what it looks like. So the colors match here, where the orange is the orange, the blue is for the centers. And you can see when migration is pretty high, there's no clear pattern. And most of our estimates were falling along the true value. Everything looks fine. However, when we start getting lower and lower migration rates, those ends start creeping up higher and higher. And what you eventually find is and these axes are different, so this is 10 times, 20 times, and 30 times greater than the true number. You can see that the ends start to get exponentially greater than the centers. So this was pretty cool, but the reviewers weren't super impressed by this. This was just some simulation. They were curious, how could this actually impact a real data set? So we went out and we found an empirical data set of lizards distributed across southern Brazil for this specific subspecies complex. So the subspecies here are labeled A through E, with each of the colors representing the different subspecies, and you can see they're broadly sampled across southern Brazil. So we thought, okay, what we'll do then is we'll subsample, where we'll keep just those groups that were far away from each other and those that were near each other. And then we'll let the phylogenetic programs estimate divergence between those and see if we can get the same kind of pattern that we're seeing in the simulations. So these were our far groups, so just grabbing those subpopulations from the ends. and then this is the near, so just grabbing those that are closer to one another. So we just reconstructed the phylogeny of those two. So this is for the near, and this is for the far. This is the subspecies that we actually subsampled, and then up here, these were just outgroups. We didn't mess with or change any of those. So as we expected, we found significant difference between divergence time estimated from the far relative to the near. The far gave us significantly greater estimated divergence times. And while that was something we kind of expected, what we didn't expect to see is that this impact actually rippled across the phylogeny. So despite the fact that we did not subsample any of the rest of these clades, they all too had deeper divergence times, indicating that when you're estimating substitution rates, that each of these are correlated to some extent. They're impacting one another. And so that was a really interesting pattern that we didn't expect to see. Okay, so spatial structure, as we just said, inflates divergence time estimation. This effect is lessened when you can actually take your samples from near where the zone of vicariance actually is, and the impacts are rippled across the tree. They impact the entire phylogeny, not just locally. Okay, so as I just showed you, like spatial structure is really important and something that we should think about. But one of the things that you'll notice is that even in that previous model, we still had some degree of random mating. And anytime that we're doing this sort of like FST comparisons where we're taking our sample sites and we're treating them as populations to then measure FST between them, we're assuming those populations are random mating, right? When in reality, they're not. If you were to start at any one of our sample sites and just walk along the beach, you would see a continuous distribution of amphipods across the entire beach, right? There's no real discrete populations here. And by doing that, by subsampling into these discrete populations, we're actually losing information at the individual level, right? And so this is an individual based where you're actually letting the individual pairwise divergence speak more than trying to subset it into a subpopulation. So thinking about that, I got really interested in these spatial continuous models and how we could apply them to these data sets to try to get rid of all of the artificial discretization we were doing. And you can't really dig into spatial popgen models without running into Sewell Wright and Gustav Maleko, who in the 1940s derived this important concept of neighborhood size. And again, this is to replace the dean, right? So instead of having subpopulations or deans, we're now treating the individual as the unit of spatial structure. So neighborhood size is defined as 4 times the mathematical constant pi, times rho, which is the population density, and sigma is the average dispersal distance. So what does this actually mean? So if you take just a little pretend plane with some hypothetical cartoon amphipods, and then with our orange one here as the focal, the circle around that focal individual is 2 times the dispersal distance, and so all the individuals that fall within that circle represent the individuals in its neighborhood. And you can think about this as the number of potential mates that any individual might have per generation, because those outside of that circle are beyond its average dispersal distance, and it wouldn't run into them in its average lifetime. Who cares? Why does this matter? What does neighborhood size actually tell us about a population? Well, one of the ways in which it's really important is that it tells us how much inbreeding a population is expected to have. So what these two figures are showing are observed heterozygosity and the inbreeding coefficient. Along the x-axis is neighborhood size, and for all of these points, they have exactly the same total population size, right? And then the dashed line represents random mating expectations. So you can see at lower neighborhood sizes, despite having the exact same total population size, they have much less observed heterozygosity and much higher inbreeding relative to random mating expectations. So in continuous populations, the total population size is not what's going to be dictating the amount of inbreeding, it's neighborhood size. Okay, so with recognizing the importance of neighborhood size and kind of what it means conceptually, they derive the probability of any 2 individuals in the population being related to one another like so. And I've plotted this expression on this graph here with the different colors representing neighborhood size from high or from low to very high. And you can see that this probability of being related goes from 0 to 1, which is useful in that we can see it's a continuous distribution. But in reality, no individual is absolutely has 0 probability of being related, because everybody in the population is related to some common ancestor, right? And the reason that we get this problem is because they're considering an infinite space, right? Where no range is actually an infinite space, it has discrete edges. So there exists some equilibrium rate at which everyone is related. And so these are some simulations showing low dispersal at one end and high dispersal at the other. And then on the y-axis is just how related you are. So you can see across the dispersal distances, you initially have a lot of relatedness. And then it kind of starts to decay and it starts to flatten out. Right? And what that means is that there comes a point in the spatial population pedigree where distance stops to be it ceases to be meaningful. Right, where the pairwise relatedness between you is no longer dictated by distance. So to get kind of an understanding on this, let's consider on this side the actual spatial genetic pedigree. And then over here is just the same thing, but in an IBD curve, to kind of show you how the IBD curve is capturing things that are actually happening in the spatial pedigree. So the spatial pedigree has two phases, the scattering phase, which is where the actual distance to any individual matters, where you are more related as if you are closer to one another, whereas the collecting phase represents the transition to a random mating expectation. To understand this, you can imagine if you were to cut off this part and just focus here on the blue, on the red, the blue, and the yellow, this is what random mating looks like, right? Where distance is no longer telling you anything about relatedness, whereas in this part of the tree, distance is absolutely telling you something. So importantly, recognizing that the IBD curve is capturing both of these processes, we can then partition the tree and estimate two of the quantities we might be interested in at the same time. So down here, again, is from the Wright-Mallico formula I just showed you of the probability of being related is being determined by distance. But then up here, this is just the equilibrium rate at which everyone is related. And you might recognize 4 times n sub e of mu is the equilibrium pi, right? That's the rate at which when a population is at equilibrium, everybody is expected to be related. Okay, so what we did then is just extended the Wright-Mallico model to consider these two phases of the tree. So first, we have the probability of being related, which is just the Wright-Mallico formula. So, and we transition this into the probability of being homozygous instead of just the probability of being related. And so the probability that you're related here, this is in the scattering phase, and then the second term just represents the background level of homozygosity, right? So again, this is being captured by the expected amount of diversity within the population. So now we can see we scale, instead of from 0 to 1, it scales down to some equilibrium value that everyone's related. Okay, so we implemented this model in RStan, where the user inputs two matrices, a pi matrix and a pairwise geographic distance matrix. We then have a series of parameters that are estimated. Our likelihood function is Wishart distributed. Then we run an MCMC, and the outputs are then estimates of neighborhood size and estimates of the long-term effective population size. Okay, so what I'm going to show you now is the results from some simulations across a range of different parameters to just kind of test how the model performs. So starting for each one of these panels are going to be dispersal distance at low to high, and then you're going to see colors that represent population density from low to high. The x-axis is going to be the theoretical neighborhood size, and then the y-axis is going to be what we actually estimated, and you're going to see a dotted line that represents the one-to-one. So across most of these, we found a pretty good fit between what we're actually estimating and the theoretical expectations. At sort of the lower end, we slightly overestimate, and then down here at the higher end, we slightly underestimate. But for most of the values in between, we see that the estimates fall along that one-to-one. So this is showing you the same thing, but except for effective population size as opposed to neighborhood size. So again, the x-axis is the theoretical, y-axis is the estimated, and you can see for most of the estimates we fall along the one-to-one. Again, with some of these still falling a little bit below, so we're slightly underestimating the long-term effective size compared to the neighborhood size. So then we wanted to apply this to an empirical data set, so we chunked this at the Red Sea clownfish, which was sampled all throughout here. And this is the highest posterior density for the estimated neighborhood size, which ranged from 51 to 53. We didn't really have a good intuition about what the neighborhood size of a clownfish really was. So we dug around in the literature and we found a population survey in 2016 that did a 200 by 50 meter transect in which they just collected individuals across several census years. And they estimated that there were around 52 individuals per that transect. So we thought that we were doing a pretty good job if we were matching sort of what the population census data had Okay, so amphipods in space summary. So we started off talking about origin and evolution of the hosturiid amphipods, where I sort of walked you through the great deal of things that we've learned, and also probably gave you some indication about the great deal of things we don't yet know, including like just some basic life history stuff, and their genomics is still very much in its infancy. Second, we talked about how spatial structure can influence divergence-time inference, specifically can inflate divergence-time inference. And then finally, I presented a model for evaluating patterns of relatedness in organisms using patterns of isolation by distance that we can extract from the IVD curve. So the next steps, one of the things I'd really like to do is generate more genomic resources for hosturiid amphipods, including more population-level investigation to better understand their dispersal potentials, life history strategies, etc. Okay, so with that, I want to thank all of my great collaborators, the Bradford Lab, and all the people that helped me collect. I went through a lot of trouble to ensure that I found pictures of them out in the field doing collecting, because these 4 people pick on me for not having very professional photographs and only ever giving them fieldwork photographs. So I wanted to be sure that I showed pictures of them also in the field. And yeah, with that, I will take any questions. Yes.
Hey, awesome talk.
Thank you.
I had a small question about divergence time inferences. So of course, I think the right way to do it is your approach, but there are cases when you have maybe just a few sequences and you're more interested in a broader phylogenetic scope. So I was wondering if you maybe tested or giving any thought about how to use these newer versions of the multi-species coalescence models that allow for gene flow to reduce the bias in cases where the sampling is more what people would call phylogenetic rather than population.
Yeah, so there's a few problems with like broad-scale phylogenies from the multi-species coalescent with gene flow. One is that it's just slow. The regular one without gene flow is incredibly slow, right? It's incredibly slow, and it's very limited in how many species you can even include into it. So I think one of the biggest problems with the multi-species coalescent is that it does require generally like iterating over all possible gene tree topologies, because you want to account for ILS, and then spatial structure contributes to ILS, and it actually like inflates ILS. It makes it worse. And so to also incorporate gene flow, it just is going to slow it down even further. And I've done some other stuff showing that in continuous space, you still get these sorts of problems that aren't accounted for simply by like gene flow estimates using ILS. So while I think it's better and it will improve estimates, assuming that we can find a way to make it more computationally tractable, I don't know if it's going to solve problems that emerge from continuous space, right? Because again, with just the gene flow estimate, you're still assuming some level of discretization. And so you're still missing some of that continuous structure. But it's a step in the right direction. Other questions? Yes.
We have a question from Jacob Burke. Super interesting talk. Do you think there may also be interactions among animal size, genome size, rates of genome evolution, and IVD effects on divergence times? Hard question, of course. For example, I might also expect smaller animals to have faster rates of molecular evolution, which presumably interacts with the IVD effects you're detecting on a macroevolution scale.
Okay, I'm going to start at the end, and then you might have to read the beginning for me again. So in terms of the rate of molecular evolution, these are annual reproducers. So despite being very small, the rate of molecular evolution shouldn't be any slower than any other annual organism. In terms of the relationship between like IVD and genome size, I haven't thought a lot about that. That's a really interesting thing that would be worth looking into. In terms of like the correlations between like body size, genome size, temperature, etc., all these other things, yeah, a lot of them are like sort of conflated with each other, and they're like autocorrelated, and so it's hard to know. So like with ectotherms, you always find bigger organisms at higher latitudes and at deeper waters, right? Because like cooler it is with temperature, then the bigger the body size is. So like separating the relationship between just being bigger and like what's actually driving genome size is kind of a difficult thing to do. Yeah, so I think first we have to find where these patterns are, like where the correlations actually exist before we can even begin to start trying to tease them out. I think, yeah, and hopefully just by digging in a little bit into what's actually driving genome sizes, so like looking at repetitive content, looking at transposable element evolution, that sort of thing, will help us get an idea about sort of like the population genetic environment that allows genome sizes to proliferate, right? So like if population size and body size are related, such that the bigger you are, the smaller your population size is, then you might expect that transposable elements can proliferate more because drift is more potent, right? So like a lot of these things are like interrelated to each other, and so it's difficult to tease all of them apart. I don't know if that was a very clear answer. Any other questions? All right, thanks so much. Oh, yeah, go ahead.
Oh, I was just going to ask, so you found a separation in the amphipods like 100 kilometers?
Mm-hmm.
Is there like a geographic reason for that? And is there, do you see that in other taxa that would be like a reason?
You do see it in other, like just gammarids can be in the same lake and have, like, so like obviously gene flow you would expect to be very easy between them, and they can have significant differences. In terms of like the southern Texas coast, there is nothing. There is nothing that an amphipod could not very easily cross. I have some like harebrained idea that it could have something to do with like coastal replenishment and people driving along the southern coast that has stopped, because like you don't find them along South Padre. They just vanish because of cars. If you don't know this, in Texas, you can drive your car on the beach. And they drive their big trucks all along the beach in South Padre, and they destroy it, and you don't find amphipods there. It's not really enough to cause that big of a divergence, but it might contribute. Yeah. All right, thanks so much.